Geotrichum species is a yeast-like fungus found as normal microbiota in the lungs, blood, mouth, intestines, and skin in most humans. Yet for immunocompromised patients, it can cause disease or even dissemination. It usually grows rapidly and matures within 5 days. Geotrichum produces arthroconidia, which are asexual spores formed by breaking up of a hyphae at the point of separation. The resulting cell appears rectangular or barrel-shaped. The growth of geotrichum starts with long filaments known as hyphae being produced out of a spore. The hyphae are septate or divided, which then further divide into arthrospores. Geotrichum can reproduce asexually, which means that the arthrospores break off and continue to replicate. Geotrichum infections can cause serious illness in immunocompromised individuals, including encephalitis, endocarditis, and pulmonary infections. To treat effectively, Geotrichum must be differentiated from other similar yeast-like organisms or even filamentous organisms. A series of tests done in the Mycology lab can be used to identify Geotrichum. Initially, a technician may believe that the growth in the media is filamentous, so a tape prep with lactophenol cotton blue stain may be done to rule out filamentous organism. After initial review of the slide at 40x, the technician may see arthroconidia and hyphae with no presence of blastoconidia, differentiating it from another yeast-like organism known as trichosporon. Coccidioides species is a filamentous organism that also produces arthroconidia. However, it has the presence of disjuncture cells, which Geotrichum species does not. A wet mount at this step would also be appropriate for initial examination. Here is what a wet mount of Geotrichum species would look like at 40x. The technician would then proceed to identify the organism through MALDI-TOF, an automated method that ionizes particles of the sample with a laser, which are then separated by mass and charge. The sample is spotted on a MALDI target plate and loaded on the analyzer. Within minutes, an identification of the organism may be produced. If MALDI ID is unable to identify the organism, then setting up additional media may be deemed helpful. Mycocell agar, a media with the active ingredient cyclohexamide, inhibits faster-growing saprophytic fungi, and the chloramphenicol inhibits bacteria. Inoculated and left at 30 degrees Celsius will show growth on the media within 48 hours, which in turn would rule out similar-looking organisms such as basidiomycetes. A last effort to rule out non-Geotrichum species would be to inoculate the organism on urea agar slant to determine if it contains the enzyme urease. Geotrichum candidum, for example, will be urease negative, whereas trichosporin species would be urease positive. Urea agar contains urea and phenol red as the pH indicator. Organisms capable of hydrolyzing urea form ammonia as a byproduct, thus turning the media alkaline. The pH indicator will turn the pale yellow media to pink-red in color. This is not a perfect method for identification, as certain species of Geotrichum can be urease positive, so this most likely would be helpful as a supplementary test.